Assalamu alaikum. This is lesson number 4 in our A2 level biology series. And today we are going to discuss about homeostasis in plants. So these are the learning objectives as per CIE syllabus. You have to explain that stomata have daily rhythms of opening and closing and also respond to changes in environmental conditions to allow diffusion of carbon dioxide and regulate water loss by transpiration. You will also be describing the structure and function of guard cells and explain the mechanism by which they open and close stomata. And lastly, we'll talk about the role of abscisic acid in the closure of stomata during times of water stress, and the role of calcium ions as a second messenger should also be emphasized. So these are the learning objectives, and let's start then. So like animals, plants also need to maintain homeostasis. And but when we talk about regulation and homeostasis in plants, it's much simpler as compared to animals, right? In plants, homeostatic regulation is basically to maintain an adequate uptake of water and nutrients from the soil into the leaves, right? And secondly, it has to control stomatal opening, right? So that water loss is minimized and carbon dioxide is maximized, right? So these are the two homeostatic mechanisms that a plant is working for, right? So usually when plants respond to environmental conditions such as high temperature or salinity, they're actually trying to balance several conflicting demands, right? So this is what is plant regulation all about. Okay, so plants are fairly tolerant of changes in the solute concentration, right, of the extracellular fluid. So they're rather tolerant and more, provided that the solute concentration is hypotonic, right? It is hypotonic to the solute concentration inside the cells. So in this case, they would be rather tolerant, right? But if the extracellular fluid becomes hypertonic to the solute concentration of the cytoplasm, then what would happen? Water would actually diffuse out of the cytoplasm, right? And if water diffuses out of the cytoplasm, this will result in plasmolysis, right? And this will cause the cell to shrink, and potentially this will cause cell death, right? So this hypotonic situation is actually more dangerous in terms of plants. Okay, so here in diagram A, if you notice, the solute concentration in the cytoplasm is more, right? And the solution here is hypotonic outside, right? So in this case, water potential is, water will actually diffuse into the cell, right? So this is one condition. So water will move from from higher concentration to lower concentration. But in this case, if you look at diagram B, there is more solute outside. So this solution here is now hypertonic, right? So water concentration is less as compared to the cell. So water will actually come out of the cell. This will cause the cell to shrink. So you can look at the size difference as well. Okay, this can eventually lead to plasmolysis, right? So when we talk about vascular plants, the rate of movement of water, carbon dioxide, and oxygen, and this exchange between atmosphere and the internal spaces is basically regulated by the degree of opening of stomata, right? So this is actually what is maintaining the balance here. Stomata. So this is a diagrammatic view. Mostly stomata are generally abundant on the surfaces of the leaves, right? But they are mostly common on the underside here. So here you can see, here they have shown you two guard cells and stomata in between. So stomata is actually the hole, the aperture that is between the two guard cells, right? So stomatal pores in the epidermis are bounded by two highly specialized guard cells. So here also you can see, this is one guard cell here. This is another guard cell and stomata is between them, right? So this is the opening that actually maintains the exchange of carbon dioxide, oxygen, and water. And you can see one over here, right? So they're present on the surfaces, but they're more abundant on the here on the underside, right? So this is another enlarged view. So you can see cuticle, the upper epidermis, and the palisade cells. Then the bundle sheet, xylem, phloem. Then comes the lower epidermis. And here you're actually seeing the guard cells and the stomata here in the middle, right? And the same you can see here also. Okay, so let's discuss about the structure of guard cells and then we'll talk about the functions. So guard cells, these are the guard cells, which you can see here. Okay, they exist in pairs. So guard cells have three structural features, right, which explain their functions, right? So they're specialized cells. So the structure is actually related to their function. So the first feature is that they're joined at their ends in pairs. So you can see here, they are joined here, this part and this part. So they are joined at the ends, right, in pairs. Now the cell walls are thicker on the side nearest to the stomatal pore. So this side here of the cell wall is much thicker, right? So the inner side is much thicker. Okay, and this side is thinner. The outer side actually is thinner. So we'll talk about that in the other diagram also. This part is thinner, right? But the outer part here is actually thicker. And there are a band of inelastic cellulose fibers that run around each cell, right? So in it, there are bands of inelastic cellulose tissue fibers that run around each cell. So since they're inelastic, so the guard cell will not change their length or their width, right? Okay then, so let's talk about the structure of guard cells in detail. So each stoma, right, is surrounded by two guard cells. So we just saw in this previous diagram. So this is a stoma, right? And it is surrounded by two guard cells. Then what other features guard cells have? They have thick cell walls, which are facing the air outside the leaf and the stoma. So we just saw that in the diagram, right? So these are thick cell walls, okay? The one which is facing the stoma, right? And then they have thin cell walls, which are facing the adjacent epidermal cells. So here we are talking about these thin cell walls, which are facing the adjacent cell. So this is an adjacent cell, right? And this side here is thinner, facing this adjacent cell. Okay, then cellulose microfibrils arranged in bands around the cell, right? Now, cell walls have no plasmodesmata, right? So that's another structural feature. They have no plasmodesmata. Cell surface membrane is often folded and contains many channel and carrier proteins, right? So as we talk about the structure, so we'll see how this happens. But the cell surface membrane is often folded and contains many channel and carrier proteins. Now, the cytoplasm has high density of chloroplast and mitochondria, right? So, since like ATP, there will be actually proton pumps are also there, so we need energy for that. So that's the reason we need mitochondria there, right? So cytoplasm has a density of chloroplast and mitochondria. Now, chloroplast have thylakoids, right? But with few grana, okay, which is unlike mesophyll cells. Okay, mesophyll cell chloroplast. So their chloroplasts have thylakoids, but they have few grana. So a little difference between mesophyll cell chloroplasts as well. Mitochondria have many cristae, right? Then they have a small several molecules rather than one large molecule. Okay, so that's another difference here. So they have vacuoles, but they have several small vacuoles. They don't have one large vacuole, right? But this is the structure of the guard cells, right? So each stoma is surrounded by two guard cells. The thick cell walls are facing the stoma. The thin cell walls are facing the adjacent epidermal cells. They have cellulose microfibrils. Cell walls have no plasmodesmata. Cell surface membrane is often folded, contains many channel carrier proteins. Cytoplasm has high density of chloroplast and mitochondria. Now, chloroplasts have thylakoids, but a few grana. Mitochondria have many cristae and several small molecules rather than one large molecule. Vacuole, sorry. So this is the structure of a guard cell. Okay, so through stomata, actually plants are carrying out homeostasis, right? Plants need to maintain a constant environment. For example, these mesophyll cells in the leaf, they require a constant supply of carbon dioxide for photosynthesis because they have a lot of chloroplasts there, right? So that they need a constant supply of carbon dioxide for that, right? Stomata, specifically when we talk about the guard cells, what they do, they control the diffusion of gases in and out of the leaves. So for photosynthesis to take place, we need carbon dioxide. And this is where stomata comes into play, right? So this means that stomata controls the entry of carbon dioxide into the leaves, right? So that's the function of stomata here. So basically this stomatal movement, this stoma as it is moving, it is the result of changes in the turgor of the guard cells, right? So if the water flows into the guard cells by osmosis, right? If the water comes in the guard cells, their turgor increases and they expand, right? But what actually happens, since we just discussed the inner cell wall is thicker, right? And the outer one is thinner. So this relatively inelastic inner wall actually makes them bend. So their size doesn't change, right? But the inner walls actually, because they are not moving, but the outer walls can move, right? Because they're thinner. So it makes them bend and draw away from each other. This opens the pore. So if we could just compare with this diagram here, see as the turgor increases, right? So what happens? Because this one is inelastic, right? It doesn't move. But this one, because of turgor, this is thinner. So this causes these guard cells to bend, right? So they would bend here, right? So this is how they're bending because of this wall being thin. Okay, as the turgidity increases, they bend, and this is what opens the pore. Then, as they bend, the pores are open. So as the stomal aperture opens and closes, this balances the need for carbon dioxide uptake by diffusion, right? As well as the need to minimize water loss by transpiration, right? So through the opening and closing of a stoma, this is what's happening. Okay, so these are the environmental stimuli that cause the stomata to open or which cause the stomata to close. So the ones which actually cause the stomata to open are increasing light intensity, right? Because for photosynthesis to occur, we need light, right? So, and then the stoma will open, they will allow the carbon dioxide to get in for the purpose of process of photosynthesis to take place. And then the second condition in which the stoma would open is low carbon dioxide concentration in the air spaces within the leaf. So that's another reason. The cell wants more carbon dioxide for photosynthesis to occur. So these are two reasons which would cause the stoma to open. One is increasing light intensity. The second one is low carbon dioxide concentration in the air spaces within the leaf. While the environmental stimuli which cause the stomata to close are darkness, because definitely photosynthesis is not taking place in darkness, right? So stoma will remain closed. Now, when already there's high carbon dioxide concentration in the airspace within the leaf, so no need to open the stoma because the plant doesn't want any more carbon dioxide to get in, right? Low humidity, right? If there's low humidity, there is less water content outside. So plant wouldn't want the leaf which is inside it to leave actually, right? It wouldn't want the water to leave. So this is another condition in which the stoma will remain closed. High temperature, so obvious reason. And then comes the water stress, right? And so this can be a desert condition, or when the supply of water from the roots is limited, or there are high rates of transpiration. So these are the conditions in which the stoma will remain closed, right? So this is just a flowchart of what we just discussed a while back. That stoma will remain open in case of already there's more water, there is light, there is low carbon dioxide, so stoma will remain open or it will open, right? And what will happen because of the stoma being open? More carbon dioxide will get in definitely, but at the same time, water will also be lost, right? So if there's a benefit, there's a definitely a disadvantage as well. And if we talk about the stimulus in which the stoma actually remains closed, so that is if already there is low water, right? And the plant needs to conserve water, darkness, when already carbon dioxide is higher. Abscisic acid is a hormone that actually helps in stomach closure. So we'll talk about that in a while also. So all these will respond, the response would be the stoma will remain closed. And the outcome will be, since the aperture is closed, so less carbon dioxide will go out, right? And benefit is definitely more water will be retained because of all this. So here again, the advantages and disadvantages of opening and closing of a stoma. So advantage kya hai if the stoma is open during the day, right? So it leaves like the leaves actually gain carbon dioxide for photosynthesis. So that's an advantage that happens during the day, right? When the stoma is open. But what will be the disadvantage? It would actually, the leaves would lose large amounts of water by transpiration since the aperture is open, right? So carbon dioxide can diffuse in, but at the same time, water can also diffuse out. So that's a disadvantage, right? Well, the stoma is closed during the day, right? So if it is closed during the day, so it means water is retained inside the leaf, which is important in times of water stress, right? So if it is closed during the day, so this is one advantage, right? But supply of carbon dioxide decreases, so the rate of photosynthesis decreases, right? So it can also happen if it is closed during the day and the aperture is not open, and since the carbon dioxide is entering through the stoma, so supply of carbon dioxide will definitely be hindered, right? So this is one disadvantage. Okay, the thing with the stomata is that they open and close in a daily rhythm, right? Even if you keep the plant in constant light or you keep the plant in constant darkness, The daily rhythm of opening and closing of stomata continues, right? So during the day, what would happen if the stomata is open? We just discussed it would maintain an inward diffusion of carbon dioxide because it's coming in, and outward diffusion of oxygen, right? It also allows outward diffusion of water vapor and transpiration, right? So during the day, this is what's happening. Why if the stomata is closed at night? We know definitely photosynthesis cannot occur. If photosynthesis is not occurring and the stoma is also closed, so this reduces the rate of transpiration, water would not go out. It conserves water as well, right? But if you just look at this diagram, so here look at part A, they have here shown you the daily rhythm of what happens during the day and what happens during the night. So this is darkness, the darker line that they've shown. So see the daily rhythm, right? What happens throughout the day, during day and night. But here in this case, what they have done, they've just shown that this is a period of constant light. They are actually, okay, they are giving constant, but even then you see the rhythm is there. The stomata opens for longer period of time, then it closes. Even though there is light, there is no darkness. See, then it opens, closes. So the rhythm stays, but definitely the level has decreased because there's like lesser light, so lesser photosynthesis taking place. But even then you can see that the rhythm is the same. And then you see this here, they have kept it for a longer period of darkness. The darker line that you see. But even then you see, even in the darker lines, the rhythm is there. It goes, it opens, it closes, right? And then again you see, it opens and closes. So rhythm stays there, right? It persists. Even if you keep it in constant light or even if you keep the plant in constant darkness. So the rhythm stays. So this is a picture from your book, a photomicrograph of two stomata and guard cells. So you can see this is one guard cell, this is another guard cell. So here the stoma is open, here it is closed, right? So you can see here the difference. Then you can see chloroplast here as well, right? Okay, so let's watch a video about the structure and working of stomata, and then we'll continue after that.
Structure and working of stomata. In plants, most photosynthesis takes place in the leaf. Let us observe a leaf section to understand the location of stomata. The outermost layer is the waxy cuticle. Under the upper epidermal layer is a layer of palisade cells containing the chloroplasts. Beneath the palisade cells is the spongy tissue with air spaces. The stomata are located mainly in the lower epidermis. Stomata are nothing but minute pores in the lower epidermis of a leaf. Let us now observe a single stoma closely. The two bean-shaped cells enclosing the stoma are the specialized guard cells. The guard cells have a number of chloroplasts present in them. Observe that guard cells have a thin outer wall and a thick inner wall. The guard cells play a vital role by regulating the opening and closing of stoma. They control the exchange of gases between the leaf and the atmosphere. Air containing carbon dioxide and oxygen enters the plant through these openings, where it gets used in photosynthesis and respiration respectively. During the day, the stomatal pores are open and gaseous carbon dioxide enters the leaves. Waste oxygen produced by photosynthesis in the cells of the leaf interior exits through these same openings. Also, water vapor is released into the atmosphere through these pores in a process called transpiration. What regulates the opening and closing of the stomata? During the day, the guard cells gain water and become turgid. That is, they become swollen or puffed out. During the night, the guard cells become flaccid, that is, wrinkled and less rigid, making the stomata close. Thus, the guard cells play an important role in photosynthesis. The factors that regulate the opening and closing of a stoma are listed here. Biological rhythm. Stomata continue to open and close on an approximately 24-hour clock even when switched to continuous light. Intensity of light. Light causes stomata to open. The minimum light level for opening of stomata in most plants is 1 by 1000 to 1 by 30 of full sunlight, just enough to cause some net photosynthesis. Concentration of carbon dioxide. Low concentrations of carbon dioxide cause stomata to open. High carbon dioxide content causes stomata to close. If carbon dioxide-free air is blown across in darkness, stomata open. Water balance or humidity. Wilting plants close their stomata.
So the mechanism to open stomata. Guard cells open when they gain water, right? And they become turgid, right? So they gain water, they become turgid, they would open. And water actually enters the guard cells through osmosis, right? If there's a decrease in the water potential in the guard cells, right, water would actually enter the cell by osmosis, right? So if the solid concentration is more, right, so it would cause water to enter the cells by osmosis, right? Okay, then in response to light, because we just discussed the stoma would open during the day, right, and allow carbon dioxide to get in. So in response to light, ATP-powered proton pumps in the guard cell surface membranes actively transport hydrogen ions out of the guard cells, right? So this is important. So this is happening in response to light. We have ATP-powered proton pumps, which are present in the cell surface membranes. They actively transport hydrogen ions out of the guard cell, right? What happens because of that? The inside of the guard cell becomes negatively charged, right? As compared to the outside. So the inside becomes negatively charged, right? So what happens because of this? This causes the channel proteins in the guard cell surface membranes to open. And what happens because of that? This allows potassium ions to move down the electrical gradient and enter the guard cells, right? So what was happening? Hydrogen, through the proton pumps, hydrogen was actively being transported out of the cell. Because of that, channel proteins in the guard cell surface membrane open and they allow potassium ions to move down the electrical gradient and enter the guard cells, right? These potassium ions also diffuse into the guard cells down a concentration gradient, right? So, so they actually enter through electrical gradient and then enter passively through concentration gradient as well, right? So this combination of electrical gradient and concentration gradient is known as electrochemical gradient. Okay, then. Now, as the potassium ions more inside, right? So the influx of potassium ions increases the solute concentration inside the guard cells, right? So we have more positive ions, potassium ions, it will increase the solid concentration, right? What would happen because of this? The water potential inside the cells will become lower because now we have more solid concentration there. Now water will enter the guard cells by osmosis, but water will enter through the water channels, which are aquaporins in the guard cell surface membrane. So most of the water actually then enters the vacuoles, right? And then it causes them to increase in size, right? So what is happening? Hydrogen was being actively transported out. This caused the channels in the cell surface membrane to open, which allow potassium to come in, right? Potassium was also coming in passively. This influx of potassium lowers the water potential inside the cell, and this causes the water to enter through aquaporins in the cell through osmosis, right? Then most of the water enters the vacuoles. It causes them to increase in size. So because of all this, the turgor pressure of the guard cell increases. As the turgor pressure increases, what would happen? This would cause the stoma to open, right? So we discussed that the inner cell lining is thick, cell wall. It would not stretch, right? So the bands of cellulose microfibrils only allow the guard cells to increase in length, not in diameter, right? So the outer thinner wall will actually stretch. So the thin outer walls of the guard cell bend more easily than the thick inner walls. This causes the guard cells to become curved, hence opening up the stoma. So here you may see right? These are chloroplasts. There are vacuoles here as well, right? So what you notice here, that actually water is entering in. So this increases the size of the vacuoles. Cell become turgid, right? And it bends. The guard cells, they bend, right? And if you look at this diagram here, right? Look at the size of the vacuoles also. Okay, if the solid concentration is more, there's hypertonic outside. So water would actually leave because there's low water potential here right now. So water will leave the cell and this would cause plasmolysis as well. So here you can see. Okay, so we just discussed about the mechanism to open the stomata. Now we'll talk about the mechanism to close the stomata, right? When certain external stimuli are detected, right? So this leads to the closing of stomata as well, right? So when the stomata gets closed, the proton pumps in the guard cell would stop actively transporting hydrogen out of the guard cells because this is what was happening in the presence of light, right? But if it's dark now, there's no light. So these proton pumps, right, they would stop this active transport of hydrogen ions out of the guard cell. Since hydrogen is not going out, right, what would happen? The potassium ions would leave the guard cells now, right? Because they were entering because of active transport of hydrogen. Now these potassium will leave the guard cell. What has now happened? The water potential gradient is now reversed. And since the potassium is also going out, the water leaves the guard cells by osmosis, right? So just have a look at this diagram here, right? As potassium leaves, water also leaves, right? And this causes the guard cells to become flaccid, closing the stoma. So you can just see the difference here in this diagram, and then look at the difference here in this diagram. So this is a picture from your book, but just to recap, what happens, these ATP powered proton pumps are there in the cell wall, which you can see, right? So they actively transport hydrogen out of the cell, right? What happens because of the low hydrogen concentration here now and the negative charge build up, this causes the cell, right? So this causes the potassium pumps to open actually. So potassium channels open, right? Then potassium diffuses in, right? So potassium diffuses in down an electrochemical gradient. What happens because of high potassium here now, the water potential of water will become low, right? Now, as the water potential becomes low, what would happen? Water moves in through osmosis because water is more here now outside, right? So water moves in through osmosis, right? Down the water potential gradient. So you can see. And then the entry of water increases the volume of the guard cells. So they expand. So if this process keeps on happening, right? So water will keep on enter. So this will cause the guard cells to expand. So we know that the inner wall will expand more as compared to the inner one. So the outer one would expand more as compared to the inner one, right? And the cells would curve apart. So all this will cause the stoma to open, right? Okay, now let's talk about the role of abscisic acid, also known as ABA, in stomatal closure, right? So this abscisic acid or ABA is also known as a stress hormone, right? So during times of water stress, this hormone, abscisic acid or ABA, it is produced by the plants to stimulate the closing of their stomata. So obvious to the name as well. If there is water stress, definitely the plant would want to conserve water. So this is where this hormone plays a role, right? So the conditions which can cause water stress are very high temperature, right? Or reduced water supplies. Okay, then since it's a hormone, it needs a receptor. So guard cells have ABA receptors on their cell surface membranes, right? So these hormones will actually bind with the receptors. As they bind with the receptors, what happens? This inhibits the proton pumps, right? If the proton pumps are inhibited, so this would not allow the hydrogen to go out of the cell, right? So this ABA binds to the receptor, inhibits the proton pump, and thus it stops the active transport of hydrogen ions out of the guard cell, right? Besides this, besides inhibiting proton pumps, This abscisic acid also causes calcium ions to move into the cytoplasm. This is another role, right? And okay, so this also happens to the cell surface membranes, and these calcium ions actually now act as a second messenger. Okay, so their first step was to inhibit the proton pumps to actively transport hydrogen out of the cell. Secondly, they cause calcium ions to move into the cytoplasm of the guard cells through the cell surface membranes, which act as second messengers. Okay, so what these calcium ions do, right? They cause the channel proteins to open, right? So as the channel proteins open, this allows negative charged ions to leave the guard cells, right? Channel proteins are open, so negatively charged ions leave the cells, right? As the negatively charged ions leave the cell, this stimulates the opening of further channel proteins. Now, what happens through these channel proteins? Potassium ions also leave the guard cells, right? So calcium came into the cell, into the cytoplasm through the cell surface membrane, act as a second messenger, cause the channel protein to open, and first allows the negatively charged ions to leave, and after that opens another set of channel proteins which allow the potassium to move out, right? Now the calcium ions, right? One extra thing that they do is they stimulate the closing of channel proteins that allow potassium ions to enter the cell. So they would open the channel proteins that allow the potassium ions to leave the cell and close the ones which allow the potassium ions enter the cell, right? Since the ions are being lost, negative ions are lost, then the positive potassium ions are lost also. This increases the water potential of the guard cells now, right? So water potential of the guard cells is more now, and the solution outside has become hypertonic. So this causes the water to leave the guard cells by osmosis. This causes the guard cells to become flaccid, and this would lead the stomata to close. So this is the function of abscisic acid, which it plays in the stomatal closure. So here they have shown you guard cells, right? In which the proton pumps are also actively, they're transporting out hydrogen, right? Potassium channels are open, potassium is coming in, and this is causing the water to come in, causing the guard cells to become turgid, right? And the stoma to open. Now this is happening in the presence of ABA, right? This ABA has receptors on the cell surface membrane. What it does, it inhibits the proton pumps, these pumps, the ones that actually transport hydrogen out of the cell actively. They inhibit that, and they also cause calcium to move into the cytoplasm. Calcium acts as a second messenger, right? And it will open the channels that would actually cause the negative ions to go out. So you can see the chloride ion being lost, right? And it will also cause the potassium ions to go out through the channels. And it closes the channels which actually let the potassium in. So what happens, since the solute concentration is more now outside, water would also leave the cells through osmosis. And this would actually lead to the closure of the stoma. So this is a function of ABA in a stomal closure. So this is just a view of showing you that this is the cell membrane and these are the proton pumps, right? And how they actually transport hydrogen out of the guard cells into the extracellular fluid, right? And these are simply the same role of potassium ions as stomal opening and closing. So this is another video about the opening and closing of a stoma. So let's have a look.
The opening and closing of a stoma occurs by means of a potassium pump. This mechanism is also controlled internally by a plant hormone, abscisic acid or ABA. An external factor, light, causes potassium ions to move into the guard cells from the surrounding epidermal cells. Potassium ions move by active transport through specific channels in the cell surface membrane of the guard cells. This transport requires the presence of adenosine triphosphate or ATP. The increase in the concentration of potassium ions in the guard cells causes the transformation of starch into malate in the cytoplasm and a decrease in the water potential in the cells. As a result, water enters the guard cells from the surrounding epidermal cells by osmosis. This brings about a change in the shape of the guard cells and the stoma opens. In the late afternoon, when the potassium ions leave the guard cells, the malate in the cytoplasm changes back into starch, and the water potential in the guard cells increases. Water leaves the guard cells, their turgidity decreases, and the stoma closes.
Okay, so this question is from your book. They're saying abscisic acid ABA is a weak acid and its structure can be represented as ABAH, right? Since it's a weak acid, it dissociates into positively charged H ions, protons, and negatively charged ABA ions as shown. So they have shown you an equation as well, right? So the following observations have been made by the scientist. So this is the first observation. Light stimulates a proton or hydrogen ion uptake into the grana of the chloroplast, right? So we know that there are chloroplasts there in the guard cell. So if you see here, so this is what they're saying, light stimulates proton uptake, right? So we just discussed that ABAH, it dissociates into hydrogen ions and ABA negative ions. So light causes this grana to uptake hydrogen, right? Okay, and what else happens is that ABH can diffuse into and out of the chloroplast, but AB, a negative ion, cannot, right? So if it's in its normal acidic form, it can diffuse into and out of the chloroplast. You can see in this form, it can enter and leave the cell. It can diffuse in and out. But if it is in the form of NiN, in this form, it cannot enter, right? And this information is summarized in the diagram below. So you can see here, see as light, what is light stimulating? Light is causing this grana to take up this hydrogen here, right? This is a stroma. So the first part of the question says, using all the information provided, predict what happens to the pH of the stroma in the light. So now compare. This hydrogen is being taken up by the granum, right? So what do you think will be the pH of the stroma, right? It entered as a whole, but then it dissociates and hydrogen goes here in the granum, right? So what would happen to the stroma, to the pH, right? So if you compare these two, the pH of the stroma would actually increase, right? The pH of the stroma would actually increase, right? Because if you compare the stroma and then the granum, so pH here would increase. Okay, so they ask us to predict. Okay, part 2, they're saying when light shines on the chloroplast, right? When light shines on the chloroplast, right? Dissociation of ABH is stimulated, right? So when there is light, dissociation of ABH is stimulated. And now they're asking, explain why this happens. So this question is of 2 marks. So when light shines, ABH is dissociated. Explain why this happens. Okay, so what would happen? This would actually cause the proton here in the stroma to decrease because it is entering here in the granum, right? Because they're saying as light shines on the chloroplast, this dissociation is stimulated, right? So this leads to concentration of this here proton in the stroma to decrease as it enters the grana, and it shifts the equilibrium to the right. This is what it does. It shifts the equilibrium to the right. So for 2 marks. Then they're asking, explain the effects that this will have on the diffusion of ABH into or out of the chloroplast. Okay. The effect that this will have on diffusion of ABH, the entire abscisic acid, into or out of the chloroplast. So see, so as it said, ABH can diffuse into and out of the chloroplast, but ABA negative ion cannot. Since it is being dissociated here, right, and this H positive is actually entering the granum, so it will actually increase the diffusion of ABH, right? Because now concentration is less here in the cytoplasm, in the chloroplast here. So it would increase the concentration. This is what it would do, right? Okay, now it says when the mesophyll cells of the leaves become dehydrated, so we have less water now, some of the ABA is stored in the chloroplast is released. So this abscisic acid, which is stored in the chloroplast, is released into the transpiration stream in the apoplast, right? So as the mesophyll cells become dehydrated, some of this abscisic acid, it is stored in the chloroplast, it is released in transpiration in the apoplast. So if you remember from a previous discussion, ABA actually what it does, it helps in closing the stoma, right? So now they're asking, ABA travels in the apoplast pathway, right? And apoplast is through the cell wall to the guard cells. Explain why this is an advantage when the leaf is dehydrated, right? So this ABA is traveling in the apoplast pathway, and explain why this is an advantage when the leaf is dehydrated. Yeah. So since the leaf is already dehydrated, right? So what advantage will this ABA have? So see, it since it will cause the stoma to close, it will actually help in conserving water. So that is the advantage when the leaf is dehydrated, right? For 2 marks. So this question is from your book, right? And let me show you the answer key also. So here's the answer key. And this is just to show you different pathways. So the one in orange that you see here, this one is the apoplastic pathway, right? So it happens through the cell wall. And the other one is the symplastic pathway, which is happening through the cytoplasm. Just to give you an idea, I've already done this in AS, but just to recall, right? Because this ABA travels through the apoplastic pathway through the cell wall, right? So I hope you've got the concept here, and we are done with the chapter. So inshallah, hopefully, you've understood the basic topics of homeostasis. And inshallah, in the next lesson, we'll start with the new chapter. Thank you.